Begin, and I'll just start by saying I'm really happy to address this group. I wish, of course, I could be visiting in person, but I guess this is the next best thing. And so I'm going to be talking about a variety of genome analysis approaches related to precision medicine. And I'm going to be focusing really on cancer genomics and this problem of discovering the key driving genes in cancer. And I should add that my talk, you can download it if you want through the website. So you obviously all know cancer is a very important disease to study. It's a huge issue, and it's a major cause of death. And there's many different types of cancer. You probably all know, often classified by the origin of the cancer. These are some of the more common ones shown here that men and women get. Now, one thing that's kind of interesting is that you probably heard of this notion of the Precision Medicine Initiative, which is an initiative we have in the United States that was launched by previous President Barack Obama. And one thing that I kind of highlight here from his State of the Union address, he had this quote here: what if matching a cancer cure to our genetic code was just as easy? And the idea here was it really was signaling to this notion that if we really knew the exact pattern of mutations in a cancer, that could be very prescriptive in terms of treating it and understanding it and so forth. And so this, I think there's been a lot of interest since then in precision oncology, which is a sub-branch of, I guess, precision medicine. And I think to kind of introduce a little of this, I want to just talk about the notions of mutations and changes in the genome. And so first of all, we want to sort of put this in perspective. It's a bit of a numerical game to get a sense of these things. So if you look at a typical person, okay, and we kind of compare one person to another person and one person to the reference genome, often we say that, this person might differ in maybe 3 to 4 million positions. These are called SNVs, or single nucleotide variants or polymorphisms. They often have maybe 500, 600,000 indels. They have many large changes, structural variants, and this number is from the what's called the 1,000 Genomes Project, which completed a number of years ago. This number of larger events has since been revised quite a bit upwards. And most of the variants in a person are common. That means they're shared by lots of other people in the population, and only a small number of the variants are rare. But of course, when you sequence a cohort of people, you sequence people over and over, most of the new variants that you're uncovering are, of course, these rare variants. Now, these numbers, this is our germline variation, and it's useful just to keep these numbers in the back of your mind when we come to talking about somatic variation, which is the variation you have in the cancer genome. And now this is a much wider range, much more differs between people and differs between different cancers. So I'm just going to show a typical number here. So typically your somatic variation has maybe about 5,000 to 10,000 variants. Now, what's also quite useful is to break these variants up into what's called coding and non-coding variants. Those are, of course, the coding ones directly impact genes, and the non-coding ones are in the non-coding regions. As you probably know, the non-coding regions occupy the bulk of our genome. And so if you look at our germline variation, most, of course, of the non-coding, only about 20,000 hits the coding. And if you look at this somatic variant in this typical cancer, you might only have 50 main mutations in the coding regions. Now, one of the problems that people think a lot about in kind of precision oncology is really determining the key variants. And these are the variants that are really driving forward the cancer. And you can kind of think of this as a kind of where's Waldo type of problem here, where you're looking at all these variants, whether it's the 5,000 variants, or maybe some of the germline variants are important as well, and you're trying to understand which of them is really the key variant that's may be very impactful and is really positively selected in driving the tumor forward. So a lot of thought goes into how we go through all those variants and find these key ones. And part of that thought is this kind of canonical model we have of drivers and passengers in cancer. So I refer to a paper from a famous scientist, Bert Vogelstein. This is the kind of canonical model that we tend to have. And that is that a typical tumor only contains, I don't know, let's say six driver mutations. And these are identified through signals of positive selection. So they're positively selected in the group of cancerous cells. And the way we tend to find these is we look at many people that have cancer and we compare them to people that don't, or we compare it to the healthy tissue in those people. And if we see a recurrent mutation that occurs over and over again, we know that's a signal of it being positively selected and driving the cancer forward. And that's as opposed to kind of the neutral mutations, which you can think of as collateral damage in the process of the genome getting all messed up. And we tend to use the term passengers for these, okay? And so typically the thought process is we'll have thousands of passenger mutations in a typical cancer genome. And this is I would say, the dogma, and we're going to challenge this a little bit today, but we're mostly going to build on it in discussion. So as I've said to you, a key aspect to think about when you're talking about this is that the genes, protein coding regions of the genes at least, occupy only a small fraction of the genome. And so most of our genome, most of our 3 billion or so base pairs is non-coding. And so if we want to really think about all the different mutations, we really need to annotate and know what those non-coding regions are doing, because often they're controlling the genes, and there where the bulk of the mutations in cancer occur. And there's also the notion of mutational impact, and I thought I would just introduce that here in relation to sort of a transcription factor binding motif. And so the notion is that certain places in the genome, it makes a big difference if you make a mutation there. In other places, it won't make a big difference. And obviously, if you have a motif that's bound to by a transcription factor and you affect the position that's very conserved, that will be a very high impact mutation. In the coding area, you often have loss of function mutations being very highly impactful. Now, recently there was a very big project that was finished to do with cancer genomics. It's called PCOG, Pan-Cancer Analysis of Whole Genomes, and it published its collection of papers a number of years ago in Nature. And this project basically looked at the whole genome sequences of about 3,000 cancer patients. They have tumor normal pairs. And then in total, they got about 30 million somatic single nucleotide variations in their catalog. And so we're going to spend a lot of time talking about these mutations. And I should point out that this project kind of subsumed the US TCGA effort, which was kind of part of it to some degree. So one of the key things we want to do, we want to measure this mutational recurrence. And a big aspect here, it's a very technical one, is to look at the recurrence of the mutations relative to the background mutation rate. And we also want to think about how we annotate the genome and find parts that are functional. Now, part of thinking about the background mutational rate is this notion of signatures, which I'm going to introduce to you in a bit. And I'm also going to talk to you about, maybe there's some mutations that are not that impactful, but in aggregate, they have a big impact. And so we're going to maybe potentially talk about that. So here's an outline for what I want to talk to you about today. And basically, I want to talk to you about this background mutation rate and some tools that we've developed for, kind of correcting it and finding regions that are more mutated. And then I want to zoom in a little bit on talking about what we've done to annotate the non-coding genome. And then I want to talk to you a little about the signatures. And if I have time, I have this added section here. We'll see how it goes with finishing the thing on time and stuff, if we have time to talk about that. So let's begin now with the background mutation rate. So I'll just introduce the concept. So here again is looking at maybe a number of individuals that have cancer, and you can look at their mutations. And kind of naively, if you look at them, you might say, oh, there's a lot of mutations in this region, and so, oh, this might be some driver. But one problem here is that often the number of mutations you get in a particular spot in the genome, they're highly confounded by what's called the replication timing, or many genomic covariates. And so later replicating regions of the genome tend to accumulate more mutations. It's just a well-known process having to do with, I think, fewer free nucleotides. And so, of course, if you just if you look at regions of annotation and you pile up your mutations, you might think, oh, these are, maybe driving regions, and these are not, but that might not be true. And so one thing we need to do is to disentangle that covariate. And this picture here shows the tremendous variability of mutation rate in relation to replication timing. So you can see the mutation rate is shown in black, and the replication timing is shown in red, and you can see it covaries very much. And also chromatin openness, you can see that's highly correlated in an inverse way with the mutation rate. And this picture also just shows too that just the tremendous difference in mutation rate per patient that we have to take into account, and the tremendous difference between the cancers. So there's a lot of heterogeneity in the degree of mutations that we have to take into account when we sort of simply look for recurrent mutations. So we've developed a lot of ways of doing this, and one thing we've tried to do is use what's called the ENCODE data, which is this large catalog of epigenetic data related to the genome. We found out that a lot of this epigenetic data can help predicting the background mutation rate. So this graph shows how well we can predict the background mutation rate using, say, the replication timing data, then using combinations of other genomic covariates. And I think one thing that's notable here is, for instance, for this particular cancer, which is a blood cancer, we need many genomic covariates, many different types of things, such as these different genomic covariates are useful to accurately predict the background mutation rate. In this picture just shows which of these ones get the kind of largest projection onto these principal components. And you can see there's replication timing, but there's also many other sort of DNA signals or epigenetic signals are quite important. Okay, so now what do we do with the fact that we have all this heterogeneity and we have this changing background mutation rate? How do we use this in a model? So I'm just going to show you, we've spent a lot of time building different models to try to find driver mutations. And I'll just give you a sense of the different models we have. So the simplest model it's kind of a straw man, is the kind of binomial model, where you imagine there's a kind of probability for a random mutation to randomly occur, and you observe a number of mutations, and you can kind of see how likely that is with your binomial model. But this doesn't have anything to do with the background mutation rate or any of these covariates. So one of the things that we can do is that we can change the this model to allow the background mutation rate, this probability P, to vary in relation to I, the bin on the genome, okay? And then what we can also do is we can make this thing dependent on a distribution, or picked from a distribution, and the parameters of that distribution, essentially the hyperparameters here, are then dependent on various genomic covariates. And for instance, here you can think of it as replication timing and so forth. So that, and this distribution we're using for this probability, this is called a beta distribution, and this gives rise to kind of a beta binomial structure to this model. And we can do the same thing with two other distributions, a Poisson distribution, as opposed to modeling the underlying with a binomial. We can assume it's very rare and use a Poisson distribution. And then we can use another distribution called a gamma distribution. And so we have these different models that we analytically model the background mutation rate in relation to these genomic covariates, okay? Now, these are parametric models, so we have explicit parameters. So we can also imagine other models where we just shuffle the background, okay? So we can imagine just taking the background from a lot of different individuals and kind of shuffling it to kind of randomly simulate what the background is. And then we can compare that random simulation of the background relative to the observed mutations. And one of the things that we can do is we can try to shuffle the genome kind of being aware of the covariates. And these create non-parametric models that we call this particular model, this moat model that I'm going to talk about. So here I'll just tell you a little bit more about the shuffling. So we can make up different bins in the genome, and we can shuffle them, and we can think of the bins in the genome associated with different types of genomic covariates. So for instance, maybe these, the red things might represent late replicating regions, or the green things early replicating regions, or regions of open chromatin, closed chromatin, and so forth. And so we can do the permutation just within these kind of equivalence classes of similar regions in the genome. And this lets us kind of take into account the genomic covariate in relation to this shuffling. And so this kind of just shows how you do with this type of shuffling. So if you look at the overall mutation counts, how many you tend to get in a particular bin, and you look at a binomial model, you'll get this. You'll get, most bins will kind of be peaked here at the most probable value, and then you'll get a few that have more and a few that have less. And then if you look at the empirical number, it's very different, you see. And there's this big tail here that you would call as all very significant. So these seem to have many more mutations than this null has, but of course that's not very true. And if we have our beta binomial model, which is shown in sort of cyan, you can see particularly in this tail, which is the important region here, it matches much more carefully our empirical distribution. And you can see the same thing here where we use the sort of gamma Poisson model as well. Okay, so now one of the things we can do is, now that we have our model for that we can use to look at our genomic background, we can evaluate a p-value for each particular region of how significantly mutated it is, and we can find the regions that are significantly mutated, and we can see if these correspond to anything useful. And so, for instance, this is shown, just the easiest way to sort of see this right away is just a sort of Manhattan-style plot where this is the p-value we might get for a binomial test. Here's what we get for a beta binomial test. And we can also sort of observe it in a somewhat different type of fashion, where we can see the more highly mutated regions in this, if we plot it versus a uniform distribution. So now, one thing we can also do with our, one of the problems with the random shuffle model is that it's very slow. So one of the things that people spend a lot of time doing is to take the random model and compare it against the analytical models, see how well they agree, and also try to speed up the random model in a variety of different computational techniques. And I'm not going to describe them in detail, but one of the things that we were able to do was to speed these up quite significantly by using these GPUs, which are special processing units, and we could speed up this approach quite a bit. Okay, so that's finding the mutationally burdened regions. And now let me tell you a little bit about the notion of annotating the genome. And so I'll just motivate that a little bit with some simple introduction. And so where does the word annotation of the genome come from, or the phrase annotation of the genome? Well, I think it comes from what we would do with human text. So for instance, here I show two famous papers. So this is the original paper from Watson and Crick that was published quite a while ago in Nature. And here's another paper published in Nature, which is, of course, the initial sequencing of the human genome. And you can see what we might do to annotate this as well. We might color certain regions of the text in relation to their function. So this is sort of the title. This might be a sort of abstract or head sentence. Maybe these are kind of specific facts. This is an introductory paragraph. We can also highlight repeated words. We can look at the sequence exactly. And for instance, the word structure appears many times here. We can compare the words between different texts too, and so forth. And this kind of helps us understand the text. If we didn't actually know its meaning, this would be a way of approaching it sort of de novo. And so that's kind of what we do in the genome annotation world. One thing is we use functional genomics experiments, the ChIP-seq experiment, also whole genome transcription experiments and so forth to get a noisy signal over the genome. We kind of smooth that, and then we turn it into kind of like highlighter blocks that talk about what different regions of the genome, what their function might be. And we can also just simply look at the nucleotide letters in the genome, compare them with each other, and find conserved blocks that are, say, conserved between species within the human population, or just duplicated within an individual human genome. And so those are the main techniques that we use. There's many people have worked in this area of genome annotation. There's obviously a profusion of different methods people have developed. They have different tools. I'm not going to go through them all here, but I'll just mention that they broadly fall into different groups. There's tools for calling these peaks, for segmenting the genomes, well-known methods like segue, segment the genome into different blocks. There's what's called supervised methods that try to predict particular regulatory regions of the genome, for instance, enhancers and so forth. And then things that also try to link regions of the genome together to genes. And we're going to focus more on these supervised methods today. And I want to point out that one of the big issues in genome annotation is there's many information sources that give rise to information on what different bits of the genome are doing. And so, for instance, you can look at the conservation, the sequence, but you have many different assays that you can look at. And I didn't really go over the richness of the different assays. And nowadays, you even have these specialized assays called MPRA assays, which are very targeted to give a readout for a particular type of element. And I think a big challenge is to kind of integrate these all together. It's kind of like this idea of looking at the elephant from many different perspectives and to try to realize, how they all come together to say this is an elephant. And in particular, we're going to look a little bit about on these sort of massively parallel reporter assays, these type of experiments today, and how they relate to annotation. In particular, we're going to look at the star-seq assay. And this assay, it gives a kind of readout when a particular sequence is very associated with being a promoter or enhancer for a particular gene. So you sort of take the gene and that enhancer and you kind of you separate it from the genome, and you can see if that region really drives the expression of the gene, and it gives a very distinct readout. So now there's many different types of annotation of the genome. Some of the annotation is very broad. So the ENCODE project develops this very broad annotation that doesn't use many different data sources, because it can be used over many different types of cells and many different types of cellular contexts. For instance, the ENCODE project just uses a few of these well-known histone markers like H3K4ME3, H3K27 acetylation, CTCF, DNase, and so forth, to annotate the genome. And then, but also for certain specific types of cells within the ENCODE project, and more generally in sort of genomic science in detail, there's been a tremendous number of experiments done. For instance, the star-seq experiments, TF-ChIP experiments, eClip experiments, and so forth. And they give a richer variety. So sometimes we can also try to develop a deeper annotation for those types of cells. And one big issue I think that's important to think about when we develop annotation is this notion of power. You might think naively, oh, it's better to annotate more of the genome, but actually that's not really true. If you annotate kind of erroneously too many elements of the genome, or you annotate a lot of the genome, you really cut the power you have for detection, because often when you test an element, at the end you have to do a multiple test correction on all the things you're testing. And so if you're testing too many things, particularly if they're not that accurate, you'll dramatically cut your power for discovery. So you really want to have a very precise annotation as much as possible. So now let me tell you about the specific things we've done with developing genome annotation. So one of the things that we've tried to do is to develop a kind of supervised framework for genome annotation that uses one of these deep learning models. And what this does is it takes in sort of known positives from one of these star-seq experiments. And so it does a supervised model where you know a particular region is known to, say, drive gene expression and so forth, okay? And then it takes in, as input, many of these very well-known chromatin features like the H3K9 acetylation, H3K27 acetylation, and so forth. And you use the neural network to kind of put these all together and to kind of predict the output. And one of the nice things is that the particular neural network that we use here is very much sort of inspired by a kind of a vision processing thing. And so we sort of make up almost a pseudo image. You can almost see these pictures as images, okay, where we're looking at different genomic bins and they're making some pattern. And this is a kind of image recognition system, okay? And we're going to train it to recognize particular patterns. And one of the nice aspects of our system is that it will tell us at the end which of the features are most important for this type of prediction, and maybe also help refine this prediction to an even more precise region than even output by the training data. So this training data that we're using from the star-seq experiment is kind of very, you might say, is weak training data. It's not that precise and so forth, and you can actually make it more precise through application of this network. And in particular, we're using this computational procedure we call GradCam, or it's called GradCam procedure. And basically, what this procedure does is it tracks back from the actual prediction to the region in the image that's really giving rise to the strongest signal. So for instance, if you're using it on a visual processing task to say differentiate a dog versus a cat, the task at hand is to say what's in the image, dog or cat. But this procedure kind of traces the gradients back through the different layers of the neural network to say these pixels here were the ones that were really the most useful in identifying the dog, and these pixels here were those identifying the cat. And likewise, we can do the same thing now where we can find which exact pixels are important to identify or which particular types of regions are useful for identifying, say, an enhancer and so forth, which regions in a genomic window. And then with this procedure, of course, we can, there's a variety of methods of assessing its accuracy. We can do what's called cross-validated training, and you can look at our validation accuracy, our ROC score, our PR score, and so forth. And then I think you can also just look at the results of the prediction. So this is what type of prediction you get. These are the actual signals that you have on the genome. And you can see that there's a very peaked DNA signal. You can also see the K27 signal has a kind of double dip, but there's a particular structure to these signals. And then of course you have negatives on the genome as well that we're using. And then we can compare how our model does to very left out specific validation, targeted validation on, say, mouse transgenic models. And this shows how it does relative to another well-known program called the matched filter program. And then here, this shows in a particular genomic window where there's a particular candidate enhancers in this window, this shows the kind of like the importance score around those enhancers. And this allows you to kind of refine exactly where they are, so you can kind of cut them to be very precisely related to the importance score. So you end up with a more precise annotation, not as large of an annotation as you might get from the original training data that we started with. And then this just shows how you can validate this in a sense by looking at how conserved these regions are. They're, after you do the refinement, they become a little bit more conserved in certain ways. And they, after you do the refinement, they occupy less genomic regions, but they still overlap more with, say, TSSs and so forth, which is, gives you a sense that the refinement process is giving rise to more accurate regions. So that was ways of finding enhancers. Now I'm going to tell you a little bit about ways we have of looking for splice sites, which are often right outside of genes, regions where splice binding proteins, and how we predict alternative splicing. And a little bit of the background here, what people increasingly believe is that splicing happens co-transcriptionally. It happens while the gene is transcribed as opposed to post-transcriptionally. And that means the actual context on the DNA, the specific marks on the DNA are potentially quite important for determining how things are spliced, in particular, looking at the methylation state or a lot of these histone markers and so forth. And when we think about diseases, I think it's also important to think about sometimes altered epigenetics gives rise to altered splicing and altered transcripts, and that's often to do with this co-transcriptional splicing, where you'll have the altered epigenetic marks really give rise to different isoforms. And here's some particular papers on this that have looked at this particular connection. So our idea was, of course, to build some sort of computational model that would predict the splicing from the epigenetic state and also from the sequence. And it's motivated by the fact that it's this we believe in this co-transcriptional nature to the splicing, and we also want to almost just mimic the process of how in our model how the polymerase works, how it goes from the 5' to the 3'. Our model is going to kind of build up information that way, okay? And we're going to really take into account all the different chromatin features and in this particular model. So we want our model to be very interpretable. The word I think people use is an interpretable machine learning model. And so we're going to apply what's called a recurrent neural network, which is a somewhat different type of architecture than I just showed you a second ago. And let me tell you about this recurrent neural network, how it looks. So here's again the process of splicing. Here's our DNA. And here we think of the polymerase coming through. And here are all the different epigenetic marks we might imagine on the DNA. And now what we're going to do is we're going to think about kind of encoding this in a system. And so what we're going to have is you can think of it as kind of like a bunch of grids, right? Where we're going to have for each nucleotide, we're going to encode what that nucleotide is. And we can use this one hot encoding for that. And then we're also going to encode in another grid the chromatin marks at that position. And then we're going to use this recurrent neural network model. And how does this model work? Well, what it does is it takes the column here of data we have, okay, and it feeds it into the model, and then it gives an output, but it also takes the result here and feeds it into the next model. So it's kind of goes on, and then the next model, you have the input from the column, what was fed in last, and then the output, and then that's also fed into the next model and so forth. And so this is this kind of recurrent architecture here, you can see, which has almost a temporal progression to it. And each of the units I'm talking about, I'm not going to describe them in detail, but they are what's called this LSTM model. And one of the, so we have our output, our input, the pass to the next cell state, pass from the previous state. And the key idea is that these allow some form of memory to be formed, where you can remember what was upstream a little bit. And here's a little bit of a notion of this idea of memory, where you can have a kind of very short-term dependence, where if you're, say, parsing a sentence and looking for the word apple, Bob is eating an apple, that's within that particular thing. But you can also have a longer-term dependence, and that's through all of these connections, and that will allow us to look at how the epigenetics ways upstream. Now it's only upstream now, right? Because we're going in this particular direction, are maybe influencing something about the polymerase. But that makes sense because that's how the polymerase works, right? So we're going to allow the upstream context to influence our kind of downstream prediction. And here's just kind of a little bit more of the data that you get from looking at this. So here we have some an exon that's here are the splice sites, and we can look at the epigenetic profiles at the sites, and you can see that they're very marked, very different. If we aggregate the epigenetic signals, we can see this particular mark, this K36, has a very distinct quality around the splice site. And so we incorporated this into what our model we call Esperin model, which has this recurrent neural network, and then the results of the network are kind of put together, in another layer, and it predicts if something's spliced or not. And then just very quickly, we can train and test our model, and get some notion of how accurate it is in predicting things. And it's a fairly accurate model. It gets the ROC values of about 0.9 and so forth. And then what's, I think, quite important is we can also evaluate by leaving out different features. We can evaluate which features have the biggest impact. And so this K36 feature or epigenetic mark is particularly useful. There's, of course, the DNA sequence are very important, but we can evaluate the importance of all of the different features. And then we can, of course, compare this model to other models that people have developed using different approaches. I won't go into the detail, and we think we do very well with our particular model. So that's the annotating the non-coding genome. Now let me tell you a little bit about the signatures. So a little background on signatures. So this is more on the background mutational rate that you have in the genome. So now if you drill a little bit more, the background mutation process is very complicated, actually. So there's the genomic covariates that we talked about, but there's also the notion that depending on the mutational processes active, certain combinations of nucleotides will be more likely to be mutated or less likely to be mutated. Okay, so for instance, if you look at triplets of nucleotides, so here we have ACA, ACC, and so forth, and we look at how often in just background mutations you'll get a C to A mutation, that might be shown here for all the different nucleotides. It's fairly rare, but you can see C to T mutations are fairly common, as you probably know, but they're also particularly common for particular contexts. We have TCA, TCT, and so forth. And so this gives rise to kind of a signature. So you can think of all of these different triplets, and the amount of mutations you have is kind of a signature of a particular type of mutation. Now, what happens in a cancer situation is we have an observed mutational profile, okay? And what we want to do is we actually want to decompose this into some basic underlying signatures. And so people have the notion that some of these signatures are particularly related to underlying biological processes. So there's a signature that's maybe related to the mutational damage you have from sunlight, or the mutational damage you have from cigarette smoke, and so forth. And so people have developed the notion of particular mutational signatures as being associated with different mutational processes. And then there's the notion of taking the observed mutational spectra that we observe and decomposing it into these signatures. And that gives a sense of the types of not only the background mutation rate, but the underlying background mutational processes that are active in the genome. Now, this decomposition process, you can often think of as a sort of a matrix multiplication where you have your vector of mutational probabilities, you have your signatures, each of your different signatures, and then you have a kind of weight matrix where you're going to weight each of these signatures, okay? And so the goal often is to find these weights and to sort of explain the observed mutational spectra as a sum of them. And let me tell you a little bit more about the mutational spectra. So here is the mutational spectra that we actually observe, but think about it for a second. The mutational spectra that we observe is going to differentially impact different types of mutations. So for instance, this is the type of spectra that you have for, say, loss of function mutations within a coding regions of the genome. And you can see that for instance, because of the specific structure of the codons, these C to Ts and C to As will be differential there. So if you have mutational processes that give rise to lots of these types of mutations or these types, they'll preferentially give rise to loss of function events. And likewise, when we look at the different transcription factor binding motifs, so here's the motifs for instance, one transcription factor called i0tau21, another one EWSR, they're differentially affected by different signatures and different mutational processes. And some mutational processes will impact particular binding sites more than the others because of the way the binding site's particular triplet structure. And so what we can do is we can actually look at the signatures that are active in a particular cancer. I think this is in kidney cancer, okay? And we can decompose the signatures that are active. But then what we can do is we can now, this is all the mutations, this is individual patients, all the mutations that are involved. And for instance, this is signature 5. Well, we can say, look at all the mutations that are, say, low impact. And you can see that signature 5 has a smaller share of that. And then we can look at all the mutations that are loss of function. And again, you can see signature 1 has a bigger role there and signature 5 is smaller. So the mutational processes are very important in terms of what the types of mutations we observe and the impact of those mutations. And this is a kind of overall picture that shows in the PCOG data set all the different types of cancer, all the different types of transcription factor binding motifs, and these have different signatures in them, and you can see that the mutational processes just in the background differentially tend to impact on different types of transcription factors. And so that's actually kind of important because you can just see purely in terms of the background mutational processes, it will affect different parts of the transcriptional regulatory subnetwork. And here, this is the sort of similar picture, but now I've shown it in terms of signatures. So here's all the different signatures. Here's all the different cancers. And you can see that some of the signatures obviously are present differentially in some of the cancers, and they have a differential impact on the high and low impact mutations. Very, very different in the different cancers. And then I'm finally going to tell you a little bit about the work we've done to kind of do fitting of the signature. So now this is, of course, a very technical problem, and I'm going to drill a little bit into the technical stuff that we think about when we address this. And so the question to remember is, again, we have these known signatures that are related to particular mutation processes, aging, smoking, sunlight. Here's our observed distribution. We want to fit the signatures represented by X times the weights, okay? And we can do a linear regression and so forth to do that. We can also think about these signatures as coming from a kind of multinomial probability distribution, okay? And that's important because the more counts we have in that multinomial distribution, the more surety we're going to have on something, and we want to take that into account. And so this is our particular contribution here. Previously, people have really thought very much in terms of determining a likelihood by fitting the signature times the weight matrix to the observed mutation. So given the signatures in the weight matrix, how likely are we to observe this may be multiplied by a particular prior on the signatures. We've developed a way of separating this multiplication or this into first determining how likely you're to have a particular multinomial probability distribution, and then seeing how likely this multinomial probability distribution, given the number of counts, will give rise to the observed sequence of signatures. And so this is a somewhat more accurate way to think about this problem. And to determine it, you have to kind of optimize your determination of your the parameters. We have this kind of biconvex type of problem that we alternate between two different steps to solve it. And then we also introduced the notion that when we fit the things, we don't want to allow them, we don't want all signatures to kind of be able to contribute just a small amount. We really have the notion of only a small number of signatures being present in a given cancer. So we want to kind of regularize the fit, and we want to penalize signatures that have only a very small loading. And we can do that with a kind of what's called a lasso model, which penalizes things that have a very small coefficient. And so this kind of shows how our procedure does. This is how it compares to a procedure that doesn't have this penalization and so forth. And you can see that if you look at all the different signatures, and these are all different cancer types, you can see there's a much greater spreading out of the weights without the penalization, as obviously. And you can also see the effects of priors in doing this just kind of graphically. And then we can more analytically see, well, if we look at the fraction of mutations that we can fit with our signatures, we can look at that relative to the number of mutation counts. And obviously, as the mutation counts fall down, we can fit, we abstain more and more. We can't fit our model. And but what's interesting is our models, the ones that we fit, still agree very well with known gold standard. And so the idea is we have some way of very precisely knowing which mutational processes are maybe active in a particular cancer, and we abstain from calling things where we don't have the information. So I think we're probably at time here. So I'm just going to summarize and then take some questions. And I'll just leave out this grayed out part, which is there if I talk too quickly, and I would always have something to entertain you with at the end. So let me tell you now, let me summarize just what we've talked about. I'm just going to zoom through this last bit. And so we talked about genome analysis for precision medicine, and we've talked about a lot of things that are really relevant to finding the key driving mutations in cancer, kind of sub-problems there. And so first we introduced this driver-passenger model. We introduced the notion of the mutational impact, also this PCOG data set. We talked a lot about the notion of the background mutations, how we have to correct for them with the many different covariates. And then we talked about the different approaches we have that take into account these background corrected things. We have this parametric models like Larva and Nimbus that explicitly look at the genomic covariates. And then we have these shuffling models, the MOAT, that sort of shuffles but does take into account the covariates as well. Then we looked at this issue of the annotation. And I think one of the key issues here is developing an annotation that's very compact, that's a high power and accurate annotation. And here we talked about integrating many assays and how There's a lot of new assays around, such as this StarSeq experiment that really gives very good information on particular genomic features. And then we looked at the how we found the enhancers. We have a neural network method here. One of the key aspects is it uses this GradCam procedure to find the regions around enhancers that are contributing most to the prediction, and that allows us to refine the annotation. Then we talked about finding splice sites and predicting splicing. Here we have another type of neural network, a recurrent neural network that kind of sort of mimics the process in a sense of transcription. And we have a lot of the epigenetic features that are giving rise to a fairly accurate prediction. And then we talked a lot about looking at signatures. We introduced the notion of mutational signatures, how the signatures differentially impact different transcriptional regulatory subnetworks, different transcription factors. They differentially give rise to impactful versus non-impactful mutations in different cancers. And then we talked about how we fit the signatures with this particular procedure we have called siglasso, and how it it's regularized and also takes into account mutation counts. And with that, I'll thank you very much for your attention. So I'll just also acknowledge obviously the people that worked on this very much. So a lot of this work was really developed by associate research scientist in my lab, Jing Zhang, who has now moved on to a faculty position at Irvine. And she led this ENCODEC that looked at the ENCODE in cancer or the covariates, and also worked a lot with this, what we call DECODE, this method for finding enhancers, with a very talented undergraduate, Flynn Chen. Also she worked with this person, Dong-Hoon Lee, who's a very talented graduate student who developed the ESPRIN model. And with Lucas Lachowski, another talented graduate student who developed the LARVA and MOAT models. The siglasso model was developed by a very talented graduate student, Chantal Lee, who's subsequently moved on to a postdoc. And with that, I'll thank you very much.